Hello. So we continue to do the survey of fungi module. Today's agenda will be to see PEZIZA. So as usual, before we move on to today's agenda, let us see the route map of fungi module. Under Zygomycota, we have completed Rhizopus and Mucor. Then under Ascomycota, Saccharomyces, Aspergillus, Penicillium, Neurospora and Claviceps has been completed. And today we will be seeing PEZIZA and we will be finishing Ascomycota. Then we will be moving to Basidiomycota, of which we have to see Puccinia, Agaricus and Ustilago as the three life cycles. So among the fungi-like organisms, phylum Oomycota. Within that we have to see the life cycle of Phytophthora, then Albugo. Then we will be seeing lichens, mycorrhiza, fungal toxins like general topics. Coming to today's agenda, that is PEZIZA fungi. It is commonly known as cup fungi. It is commonly known as what? Cup fungi. Why? Because its fruiting body, which will be containing the ascospores, asci, etc., is a cup-shaped structure. Okay. And whenever you see this particular PEZIZA, you normally see the images like this, right? Like just you are seeing this cup-shaped structure. You are not seeing any mycelia. Are you seeing any mycelia surrounding it? You are not seeing any mycelia. Why? Because this mycelia is under the substratum. Okay, it is beneath the surface of the ground. Okay, only the projected surface will be this fruiting body called apothecium. This is the apothecium, which is the ascocarp or fruiting body of this particular Ascomycota genus called PEZIZA. Okay, and we will be seeing the structure of this apothecium, etc., in the upcoming slides. Okay. So coming to PEZIZA's description, it's a large genus, more than 100 species is there. It's a saprophytic fungi. It will be feeding on dead and decayed matter, on forest, trees, etc., which are decaying. We can see this. So yeah, then coming to its systematic position, domain Eukaryota, within which all true fungi comes. Then kingdom Mycota or fungi, division or phylum Ascomycota, class Pezizomycetes, then order PEZIZALIS, family PEZIZAceae. Then there are around genus is PEZIZA. Then around 100 species are there within this. So this is the easy to draw figure for this particular apothecium, which you can make in exam. Okay, so you can see this is the outer covering. Then you can see the mycelium. At the tip of the mycelium, you will be seeing this particular asci, which is bearing 8 ascospores. In between this asci, you can see paraphyses, which are sterile hyphae. Okay, and if you see this area, this top area where this particular asci is located, that area is known as hymenium. Okay, then below area where the normal vegetative mycelia are there, that area is called hypothecium. Okay, then the surrounding wall, this area is known as exipulum. Just keep these names in the mind. Then mycelium of PEZIZA, it is actually uninucleate mycelium, which is having many septa. You can see many septa and they are uninucleate as well. See, another way of representation. Okay, then the hyphae are hidden from the view, that we already said. Okay, it is completely branched. You can see vigorously branching and each cell is uninucleate. That thing you have to keep in mind. Then reproduction, first of all, asexual and sexual reproduction are there. Asexual reproduction is by conidia and chlamydospore formation. We know what is conidia. There is a vegetative mycelium, one of the cell. Any of the cell can form an erect hyphae called conidiophore, which will be bearing conidia. This conidia will be exogenously formed. Okay, you have to use such terminologies, then only you will be getting marks. Okay, this is the language of the paper. Okay, exogenously formed spores. Okay, they are abstracted from the tips of conidiophores during the time of maturity. Okay, then each conidium germinates to form a new mycelium. Very simple, right? See, this is the fallen conidia, a germ tube. That germ tube matures into mycelium. Now coming to chlamydospores, if conditions are not favorable, as usual, the protoplasm shrinks and it attains a spherical shape covered with a thick wall. It can be seen in chain like this, or it can be seen in even singly as well. Okay, maybe only one. After that, maybe after some space it is one. Like that, it can also happen. And the thing is that these particular chlamydospores are intercalary in position. You are not seeing it towards the apex or towards the bottom, but in between, okay, intercalary in position. Each chlamydospores after falling will be germinating into new spores during the time of favorable conditions. Now coming to sexual reproduction, this is a unique thing in this particular genus called what? PEZIZA. Okay, in this PEZIZA, in sexual reproduction, what we are seeing is actually there is no sex organ. Okay, there is no antheridium or ascogonium. Okay, here you might have seen one label called antheridium or ascogonium, but this is not a representative figure of PEZIZA. I have just taken it for showing you the mycelium is uninucleate, and just to show you that two vegetative hypha will be fusing together at the time of sexual reproduction, but we won't be calling it antheridium or ascogonium. It's just two hypha that is crossing, that is fusing. I just wanted to show that, that's why I have taken this image, but there is no such spherical bodies called antheridium or ascogonium. It will be just fusion of this particular vegetative hypha. Okay, I'll show in the other figure. Yeah, it will be just somatic fusion like this, two hypha coming together and fusing. Okay, and there won't be structures like this. This just for like making you understand that this hyphae will be fusing like this. Just I have taken this image from some other genus. Okay, but understand that two hyphae that will be just fusing. So coming back, okay, one hypha of plus strain and one hypha of another strain. Actually, these are just normal hyphae only, but these belong to different mycelium. So that's why I have taken, for distinguishing for our understanding, just mentioned it as plus and minus. In nature, actually, there is no plus and minus, but the thing is that the chromosomes or the gene alleles within this hyphae and this hyphae will be different because they are coming from different mycelia. Just to show that we have assigned this plus and minus signs. Okay, now two hyphae coming together fusing, that is known as plasmogamy. There won't be fusion of nucleus now. After that, we know the rest is very straightforward thing. Okay, two hypha fused. Okay, that means two different nuclei came together. All nuclei, we know that nuclei from one particular hypha and one another hypha came together. Now from this there will be the formation of what? Ascogenous hyphae, in which there will be two nuclei each in each cell. Okay, now at the tip of this particular ascogenous hypha there will be one particular cell that will be fusing the nucleases. That is known as ascus mother cell. Now this ascus mother cell undergoes meiosis twice, then a single mitosis. So that will lead to the formation of 8 ascospores. You are getting the point, right? So such fusion will happen all over the body of mycelium. You can see here two plus and minus will fuse, here two plus and minus will fuse, here two plus and minus will fuse. This is the reason this entire fusion of fertile branches will be happening within a structure called apothecium. Okay, carp of this PEZIZA is called an apothecium. Now this structure is formed, and you will be wondering what are these structures, these cells, right? These are the cells. Actually, these are mycelial cells. For easy way of representation, we are marking it as round cells. Actually, this is the formation. Okay, mycelium. Then for easy to draw, we will be drawing it like this. Okay, this is the cells. Okay, this outer covering of cells is called exipulum. Now coming to yeah, this is another way of representation. If you want to draw, if you don't want to draw like this, you can draw like this. Just like you know that in normal times this particular mycelium is completely like having a tussled mass. Okay, it will be together like a tuft and all. So out of these two different hyphae from two different mycelia will be fusing. That is called somatogamy. If one particular hypha's two cells are fusing, that is called autogamy. Okay, that also keep in mind. Then after fusing, what will happen? Then ascogenous hyphae will be formed, which will be having two nuclei. Then at the tip of that particular cell, there will be fusion of both the nuclei. That will be causing the formation of ascogenous hyphae, which are dikaryotic, right? So this fusion is the actual drawing. This is just for representative purposes. Don't think that there is antheridium and ascogonium. There is no such bodies in PEZIZA. Now coming to the life cycle. So what will happen after that? So after the formation of like all the hypha which are fusing, that will be forming ascus mother cell, and that will be after meiosis and mitosis creating asci, okay, asci which will be having 8 ascospores, okay. What after this? Okay, so 8 ascospores are created. Now this is the thing. These 8 ascospores, that ascospores will be liberated at the time of maturity. Ascospores will be liberated. That will be one thing. Now coming to the general life cycle. One more thing. After liberating, what will happen? After the ascus is liberated, this ascus will form new thalli. Right. That's the only thing. Now coming to the general life cycle, asexual reproduction means what? Okay, at the tip of the conidia there was conidia. Conidia got liberated. Each conidia formed a new mycelium. Now coming to the sexual reproduction, don't think that, as I always tell, there is no in this session from the beginning I am telling that there is no sexual reproduction as such in the formation of fusion of ascobunium and the antheridium, but there will be this fusion of the somatic thalli. This is just for representation. Actually, don't draw in exam like this. Last mark as antheridium and ascobunium, etc. Just show the fusion of two thalli. Now the contents of each thalli will be entering into one thalli, which may be considered as female thalli. So after this, what will happen? The entire hyphae will be fusing in between. That will be forming ascus mother cells. That will be creating asci within the body called ascocarp. That is called what? Apothecium. It's a floating body which is this kind of in the bowl kind of shape, okay, or cup kind of shape. Then this is the ascus mother cell that undergot fusion, then diploid nuclei got formed, then mitosis and meiosis formed 8 ascospores that will be released from this cup fungi or cup shape. So after release, what will happen? Since it fused with the plus and minus nuclei, there will be plus type mycelia and minus type mycelia. Then again they will be fusing and such cycle will be going on like that. So this will be the structure of ascus. Within the ascus, you can see ascospores. Yeah, same only. Then sexual reproduction knot you can take. I have explained everything. One thing you have to understand that PEZIZA vesiculosa is the genus which is the species which is very common, and we will be studying this thing only. We have studied features of this thing only. This is the representative feature, representative species of this PEZIZA genus. Yeah. Okay, these things already I have explained everything. You can just write down by pausing the video. And one thing, this ascus we told within the perisacium we will be having ascus within which ascospore will be there. Now they are telling that the ascus wall is lined by a thin layer of cytoplasm which encloses a central vacuole. This is having a small layer of cytoplasm and central vacuole. Within the central vacuole you will be getting the you will be getting the ascospores. Okay, right. In the vacuole, within the vacuole lie the oval ascospores, oval shaped ascospores. Okay, then between that you can see paraphyses, which are the sterile hyphae for protection. Then what they are telling is that the rest of the apothecium, that remaining after the paraphysis and asci, the remaining area will be will be created by the mycelium, which will be forming a pseudo parenchymatous tissue which supports the hymenium. To support this hymenium at the top ascospore area, the remaining mycelia will be compacting itself as a pseudo parenchymatous tissue that will be supporting the hymenium. Okay. Now the apothecium is sessile or stalkless, or if stalk is there, it will be too short. Okay. And it will be having a size of 2 cm to several inches in diameter, and color is of pale fawn color. Pale fawn color means what? Light brown, or in some species you can see brilliant orange apothecium. This is apothecium. Now this is the flow chart. If you want to draw, I just added. If you want to see this, you can take it down. Now if you want to draw the ascocarp easily, follow this step. Okay, you are creating an excipulum, then you are creating the head of this particular ascus, then paraphysis, then slowly creating other polygonal cells, then completing it by creating longitudinal cells here as well. Then after that you will be creating the stalk of each ascus, then 8 ascospores within each asci. Then you have to mark the paraphysis, then periderm. Okay, you can see that this is periderm, the bottom area, protective layer. This is hymenium. This is Hymeneum. Below that there is Subhymeneum. This is Peridot. So with this we complete Ascomycota. Next we will start with Basidiomycota. Thank you.